Hi, and welcome to Beyond the Labcoat, where we dive into the fascinating world of scientific discovery with our Rebuilt researchers. I'm your host, Gabby, and today we have a very special guest, Mariana, a researcher studying genetic mutations in Drosophila melanogasters, or better yet, fruit flies. Mariana has been working on an intriguing project examining two chromosomal mutations, DARK82 and C33, and their impact on the mosaic eye phenotype. Today, we'll break down what this research means, the challenges faced, and where the study is headed. Mariana, welcome to the show.
Thanks for having me. I'm really excited to talk about my research.
All right, so let's start with the basics. Your research has been focusing on two mutations, DARK82 and C33. Can you give our listeners a quick rundown of what these mutations are and why they're interesting?
Sure. So these mutations were identified in a genetic screening that my lab previously did, where we uncovered a mutant from a UBA1 gene, which basically has to do with growth. It's a growth gene. And DARK82 is the gene that we were initially studying, and it's interesting because it affects the balance between red and white tissue in a Drosophila eye, which makes it look mosaic, is what we call it. So in this case, this mutation would show more red tissue than white tissue in the Drosophila eye. And then it's also suggested that because of the loss of a patch gene, which also has to do with growth, along with reduced apoptosis, for those who don't know what that is, that means basically programmed cell death, that we lead to an excessive tissue growth, so kind of the eye looks a lot bigger than usual in a Drosophila fly, and it has more red than white in its eye as well. But then we also have a secondary mutation, which is called C33, which is a little bit more complex because it's a double lethal mutation, so that means there's 2 point mutations in there. That's associated with a ROOP gene, which is one mutation, and then NUP75, which is a secondary mutation. So our goal was to understand the mutation's responsibility for the new mosaic eye phenotype we are observing. So with C33, we see more white tissue than red tissue in the mosaic eye, but with DARK82, we see the opposite, so more red than white tissue in the eye.
Wow, that sounds like a challenging puzzle to solve. What kind of methods are you using to investigate these mutations?
Yeah, so with Drosophila, you use genetic recombination and you use microscopy. So genetic recombination, for those that don't know, so our genes, these genes are a certain kind of length away from each other. So because it's a 2 point mutation, we have to isolate them. We have to look at the genes individually to see which one is driving the white tissue over red tissue mutation. But first we have to look at the difference. So I use digital microscopy to document their eye phenotypes. So basically, we are looking with flies with the mosaic eyes that we were previously talking about and straight wings. And then that would mean we have our mutation. And then to further understand it, we would have to freeze the flies because it's the most humane way to kind of look at our specimens. We put them in ethanol and then we take pictures of the eyes to know what the difference is. And then for recombination experiments, it sounds really big and fancy, but basically what happens is that you have flies in different vials, and you literally just cross them together, which just means you mate them different phenotypes together to try to understand which one's driving more. And so in this case, our goal was to isolate either mutation A, which we call NUP75, or mutation B, which is ROOP, and then we do that. One of them is closer to DARK82, so it's harder to recombine because technically they're called linked. So they're so close together, they're stuck together, that it's harder to recombine them away than mutation B, which is all the way on the other side. So that's the one we started with since it's the easier mutation to kind of get away and look at by itself.
And you're with Dr. Kagi, right?
Yes.
Oh, I was in his lab too. I was also working on Drosophila melanogasters, but this isn't about mine. This is about yours. So what did you find? Did isolating one of those mutations give you a clear answer?
Sort of, but not really. So what we found is that we were able to recombine away mutation B, which is ROOP, since it was all the way over here from NUP75 and DARK82, and it showed that it does have a similar phenotype to C33, which is again the one that has the bigger white ratio. So we inferred that it would be the key driver of the mutation, but we haven't ruled out NUP75 since we haven't been able to isolate it, and it is the secondary mutation. So we can't really necessarily pinpoint that our first mutation is the key driver.
Sounds really interesting and important. What's the next steps for your research?
So our next steps would be to obviously try to study the other secondary mutation and recombine it away. That one takes a lot more recombination work because it is so closely linked. And then once we've successfully recombinated everything and have isolated it, we will complement it with a known deficiency to create another mosaic eye and kind of get a closer look at it. And then we will take all the pictures all over again so people can see when I present kind of what the difference in the eyes look like and what mutation is more of a key driver than the other one.
Oh. It's amazing how much work goes into understanding just one small part of the genome. What has been the most exciting or even surprising part of this research for you?
I think the most exciting part is seeing how really small genetic changes can happen with such dramatic phenotypes, even though we can't visually see it. You know, we're talking about something so small as a fly that just buzzes around us, yet it has such scientific scientific impact since we do share a lot of our genes with flies. And looking at it under a microscope really makes it surreal for us. And then the complexity of C33 was another thing that was really unexpected since it's two lethal mutations that cause one phenotype. So it's kind of like a mystery and a big surprise as to which one drives it more than the other.
Now, with this project that you're working on, are you attending any conferences? And if so, are you going to present?
Mm-hmm. Yeah, so I am attending a conference. It's actually coming up in, I believe, 3 weeks.
Oh, nice.
I'm going to the Genetics Association of America. They're holding a Drosophila conference, and then that's where I'll be presenting my research. Primarily the second half of our project, which is isolating that secondary mutation that's the closest to dark 82.
So not many people know that, but there's a whole conference just about Drosophila.
Flies.
Interesting. Okay. Well, anyways, that's a great takeaway. But before we wrap up, is there any advice that you would give to any students or aspiring researchers interested in genetic studies?
I think the most important thing to remember is that you have to be patient when it comes to genetics research, especially. Like I'm working with flies. I don't know if it's going to work or not. So it's okay if everything doesn't go as planned. It's expected, you know, it's research. You don't know the answer. So don't be afraid to learn from your mistakes. And then when you get your data back, don't just kind of put yourself down and be like, oh, this didn't go as planned. Like, this sucks. Just take it as an opportunity to learn and tweak your experiment for next time. And then you have to remember that that's how every scientific discovery was made. So
That's great. Patience, yes. Okay. Mariana, thank you so much for joining us today and sharing your research. It's been a pleasure learning about your work on Drosophila mutations.
Yeah, I was really excited to share it, so thanks for having me.
And to our listeners, thank you for tuning in. If you enjoyed this episode, make sure to subscribe to Beyond the Labcoat for more deep dives into the latest scientific research. Until next time, stay curious. Oh, all right.